Today we will continue our discussion about this TrkB has been regulated by Calnexin, or the Calnexin regulate the activation of TrkB. So in the first figure, that is on the panel A, they have discussed about this effect of this inhibitor, that is Cas, where it is mentioned at Cas. So what this inhibitor does is it will prevent the alpha and beta glucosidase to trim the glucose from the carbohydrate structures that are formed in the endoplasmic reticulum, because those glucose are responsible, those trimming of those glucose are responsible for interaction with this Calnexin. You can go through the Calnexin and calreticulum cycle video to understand how this Calnexin and calreticulum cycle works. So when these inhibitors are used, then the proteins will not be able to interact with these Calnexin and calreticulum cycles. So in this case, they need to check whether what is the effect that has been observed on this TrkB. So when there is no inhibitors are used, they can see that phosphorylated TrkBs are formed when this EGF is been introduced into the cell culture medium. However, there is no effect when these BDNF are introduced. That can be seen here in the low expression and also in the high expression. And what they have seen in the TrkB is that there is some formation of this 130 kilodalton TrkB, and there is more formation of this 100 kilodalton TrkB. 100 kilodalton TrkB corresponds to the phosphorylated TrkB, and 130 kilodaltons TrkB corresponds to the TrkB that are expressed on the cell surface. And this difference is because the different glycosylations that occurs in both the cases. And these things are also same in the low expression and the high expressions TrkB results. So, and however, when they introduce these inhibitors, the Cas, so what they have seen is that there is a reduction in the phosphorylated TrkB formations, or phosphorylation that occurs at Y8116 amino acid site, even in both in the case of this low expression and high expression, compared to the case where there is no inhibitor. So this indicates that the Calnexin somehow regulates interaction of this TrkB with the Calnexin, helps in the activation of this TrkB, and the quantifications of this are shown here. In this case, we can see that the TrkB formations are high when there is no inhibitors compared to the case when there are inhibitors. However, the phosphorylated TrkBs, there is not much effect in the lower species. That can be seen here, both in the case of without Cas and plus Cas. However, in the upper species, there is an effect. There is a statistical significant difference when the Cas is not used compared to the case where the Cas is used. And this is the immunostainings results are shown here. They have seen, we can see that TrkB's formations is in overlap with this LAMP formations, both in the case of where there is no inhibitors and there are Cas inhibitors. That area has been boxed here. So this indicates that TrkB formations is in line with the LAMP formations in the cells, or intercellular positions are in line with each other. And thereafter, what it is, they use different inhibitors, that is tunecmacin, that is represented here as TM, that will prevent the N-glycosylations of the proteins, which are of the proteins of the within the cells that are being treated with this tunecmacin. So when cells are not treated with tunecmacin and has Calnexin, then they can see there is a phosphorylated Trk formations when in the presence of EGF. However, there is not much effect in the BDNF. However, when they introduce the reagent, they can see the reduction in these phosphorylations compared to the control case. And these things at high intensities also represent the similar results And for this TrkB, they can see that TrkB formations are negligible when the cells are treated with EGF at 130 kilodaltons. However, most of them are phosphorylated. However, for the case of this BDNF, they can see some non-phosphorylated TrkB at the 130 kilodalton size. And these things are similar in these high expressions, TrkB at high expressions. However, when the cells are treated with this tunecmycin, they do not see any tunecmycin, and when they are reacted with this EGF, they see reduction in this reduction in the non-phosphorylated TrkB. So it indicates that intercellulars TrkB are mostly phosphorylated when the EGF is being used. However, for BDNF, they do not see any changes, or they do not see any changes related to this TrkB. However, when the Calnexin is depleted, in that case, without any reagent, they see that there is a formation of this phosphorylated TRK at low expressions, and there is a high formation of this phosphorylated TRK at high expression. So this is in lines, results are in line. And for this TrkB, they can see no non-phosphorylated TrkB, as there is a Calnexin is not present. So the TrkB expressed on the cell surface, the medium, that is Calnexin acts as a medium. So because of the absence of that medium, the TrkB will not be able to translocate to the cell surface. This hypothesis seems to be true from this Western blot, and same things are validated using this TrkB at high expression. However, for BDNF, they do not see any changes. And for when the Calnexin is knockout and cells are treated with TM or tunecmycin, they can see that reduction in this phosphorylated LE. So this indicates that when the Calnexin are not there, and at the same time when glycosylation is also been inhibited, the phosphorylated TrkB will further goes down. And these things they have seen even in the case of this TrkB at low expressions. They do not see any non-phosphorylated TrkB because of the presence of EGF. All the TrkBs are been all or most of the TrkBs are been phosphorylated. However, for this BDNF, they can see some of this TrkB formations even in the case of with TM treatment, with tunecmycin treatment and without tunecmycin treatment. So this could be indicates that when the Calnexin is not there, because of the overcrowding of this TrkB, there could be some formations of some TrkBs are formed that are become, because of the overcrowding of the TrkB in the cells, they are become sensitive to some extent to this BDNF. And so after validating their Calnexin knockout with and without tunecmycin, they had to check a time dependent effect of this Calnexin depletion. So on this cortisol precursor cells. So in this case, when the Calnexin are present, they can check the phosphorylated TrkB at different times, and at 1 minute, 5 minutes, 15 minutes, 60 minutes, 240 minutes, and they can see that reduction in this reduction in this phosphorylated TrkB after injection of this EGF. And when they do not have any Calnexin, they can see the more formation of this TrkB, intracellular TrkB, because there is no transport medium present in this case. So cells will the TrkB will not be able to express on the cell surface. And when it comes to the TrkB, they do not see too much difference in this TrkB at 130 kilodaltons or at 100 kilodaltons, or phosphorylated or non-phosphorylated TrkB. in both the cases, that is and they check the other markers also, such as phosphorylated EGFBR, and they can see the reduction in this phosphorylated EGFBR with time, as because of the depletion of this EGF with time. And these are the survival markers that they have also checked. There is reduction in this phosphorylated MAPK pathways that can be seen. And when they check these results, quantify these Western blots in the panel F, they can see that phosphorylated TrkB compared to the normal TrkB, that is ratio of these two, they can find out that the difference between these two are statistically significant at 1 minute, 5 minute. However, for this 15, 60 and 240, the difference between higher this dark band and the light bands are statistically significant at till 1 minute and 5 minute. That is 1 minute and 5 minute. After that, the difference are reduced and are not statistically significant, both in the lower species and upper species. And for this EGFR to AKT, it is EGFR to AKT, they can see that it's statistically significant at 0 minute and 1 minute, and for this lower species and for higher species it is statistically significant for at 1 minute. So this also indicates that this EGFR will also get affected during this EGFR and AKT pathways also get disrupted during this TrkB's activation. And these are the other markers, that is phosphorylated AKT and AKT pathways, to show how the AKT's pathways are activated. And here for the lower species they do not see any statistical significance. However, for this so one thing is the difference here is between the blue color represents the results in the Calnexin knockout, the presence of Calnexin, and orange represent the results in the absence of Calnexin. So let us revisit the discussions about panel F. So this phosphorylated TrkB and TrkB for the 1 minute, that is they compared the phosphorylated TrkB at 1 minute and compared to this 1 minute. So this difference is statistically significant for 1 minute, 5 minute. However, for this 15 minute, it is not statistically significant in the lower species, and for upper species, it is statistically significant at 1 minute. So whatever I have told about F panel previously, kindly disregard that and understand it from this point. And for this EGFR, they can see they compared this EGFR with this EGFR, and they can see that for the 0 minute and 1 minute, they can see the results are statistically significant. So this indicates that in the case of this Calnexin knockout, these pathways are upregulated compared to the case where Calnexin are present. And for this phosphorylated AKT, they can see that for the lower species there is no significant upregulation, but for the higher species they can see significant upregulations at 5 minutes. After that it goes down because the EGF amounts goes down with time. So it peaks at 5 minutes. And then they decided to check the pathways that has been reported in the literature that could be activated when these cells interact with this EGF, and so they inhibit those signaling pathways using PP1 1/2 and using PP 1/2. So in this case we can see that phosphorylated TRK is present more in the presence of Calnexin knockout when this inhibitor is not present. However, when these inhibitors are present, phosphorylated TRK goes down. When they quantify these things, they can find out that this PTRK intensity goes down in the case of this Calnexin knockout in the lower species and also in the higher species. However, for this, when the inhibitors are not present, when the inhibitors are present, the difference is not statistically significant. That is what they have compared here, is that this phosphorylated TrkB, these two is statistically significant. However, when these inhibitors are present, these differences are not statistically significant. So this indicates that the targeting component of this inhibitors plays an important role in this activation of this TrkB using Calnexin. After that, they had to check the time-dependent effect of the cortical neuron for this BDNF in the presence of Calnexin and calreticulum, in the presence and absence of Calnexin. So in the case of this phosphorylated TrkB, they can see that this the amount get reduced with in the presence of Calnexins. However, when the Calnexin is not there, the amount does not increase. However, it is almost show the similar results except for the 15 minute. And this TrkB amount is also remains similar when for the Calnexin presence and Calnexin is not present. However, this phosphorylated AKT get disrupted when the Calnexin is not present, and similar for this case of MAPK kinesis pathways. When they quantify these Western blots, they can see that for the 1 minute, this phosphorylated TrkB is statistically significant in the case of this Calnexin presents compared to the case with Calnexin is absent, and similarly for this 5 minute. That is this and this comparisons. However, for this 15 minute, it is statistically significant but to a lower extent compared to this 1 and 15. However, for 60 minutes and 240 minutes, they are not statistically significant. And for this MAP kinase pathways, the difference is statistically significant at 1 minute. At 1 minute between Calnexin is present and Calnexin not present. However, but for this AKT pathway, they can see the signals are statistically significant for a longer duration of time, till from this 1 minute, at 1 minute to this 15 minute, 60 minute, and even in 240 minutes, except for this 5 minute. So this could be because of the U-shaped curve that might occur. It is initially activated, then goes down, then again activated. This indicates that all this data in general indicates that it's this Calnexin plays important roles toward TrkB activations in the presence of this EGF. However, BDF does not have too much role towards the activation of this TrkB at a lower activity levels when the cells are at lower basal conditions. And if you need to understand more about these experimental details in experimental results and how they conducted these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.